Okay, hello. This is Dr. H, and I am going to be doing hopefully, a summarized lecture on blood. This is what you guys will need to know for the lecture, exam, and then also for portions of the lab practical. So let's get rolling on this, trying to make it somewhat short and sweet. Blood, very important part of your body's physiology because it helps with transport of material. And we got blood, it's going through these blood vessels. There's all sorts of these formed elements, these types of cells that are in your bloodstream, but there's other stuff in here as well you guys are going to learn about. But these primary formed elements are things like your red blood cells and your white blood cells, and then also your platelets, these tiny little fragments in here that help with blood clotting. And you guys are going to learn more about each one of these, what are called formed elements, in this lecture. Now, blood can either be scarlet red. I probably talked to you about this by now, but if it has oxygen bound to the iron group found on red blood cells, otherwise known as erythrocytes, right? Then those red blood cells will be red, very red. Now, as they start to lose those oxygens and pick up carbon dioxides, they're going to become more and more blue, but they never lose too many oxygens, so they tend to remain red. They just kind of become a darker red, almost purple blue color. Volume of the blood, please know these values. Men, 5 to 6 liters, females, 4 to 5 liters. Why the difference? Because men tend to be larger. This just has to do with mass. It's homeostasis. If the body is larger, it needs more blood. Whew. Total body mass. Blood counts for quite a bit of your body weight. Now, what's in blood? If you take somebody's blood, a patient's blood up here, and you put it into a tube that disallows for platelet aggregation, disallows for clotting, right? It's called a heparinized tube, or they use other things that stop clotting. And then you spin that blood down, then the heavy things go to the bottom. Well, these erythrocytes, these red blood cells, have iron on them. Therefore, they're very heavy. They're going to go to the bottom of the tube. And these red blood cells end up packing together. You're going to see a term called packed cell volume. The packed cells are the erythrocytes, and they tend to account for approximately 44% of the whole blood. Now, what that 44% means, that if you look at the total volume of blood in the body or in this tube, about 44% of the blood is comprised of erythrocytes. There are a lot. We're going to see a slide in that just a second. These play a role in transportation, not just of oxygen, not just of CO2, but many other things as well. So now, if you look at these spin components, there's a very small value, well less than 1%, negligible total percentage on the total blood volume. It's called the Buffy coat. Buffy coat has the rest of those formed elements that I talked about in the first slide. The thrombocytes, otherwise known as platelets, these help with blood clotting. And the leukocytes, also known as white blood cells, these are your immune system, and we'll talk about each one of these: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. But on this slide, you guys can start to study the names of each one of the white blood cells, and you can study them from the order of the most abundant to the least abundant. Neutrophils are the most abundant. That's going to have to do with their function. We'll talk about that in just a minute. Neutrophils are the most abundant, and they account for 50 to 70% of all of your white blood cells. And this is a value you need to know. You need to know. For the lab practical, I will be asking you guys case study questions that says a patient comes through the door and their neutrophil value is 78%. What's going on? Well, it has to do with the function of neutrophils. Again, you'll learn that in a minute. Well, probably a little bit longer than a minute, but you'll learn that. So you must know these values, and you must know what's normal, and therefore you'll be able to tell me what's abnormal. I hope that's clear. Now, that only accounts for, again, 44-ish% of the blood. So what's going on with the rest of the blood? Well, this is called plasma. And plasma is mostly water, but there are components in your plasma that you must know for the rest of the semester. A big chunk of your plasma is made up of proteins. Proteins. 90% of all blood proteins are made by, always the bridesmaid, never the bride, the liver, right? So things like albumins and globulins and fibrinogen and transferrin, and there's all sorts of proteins that are found in your bloodstream. We're going to learn about angiotensinogen and so forth, so on. And there are a lot of proteins. Most of them act as carrier proteins to something. But this most abundant protein is one I will test on multiple times, albumin. Albumins are your primary osmotic regulator of your blood. Remember what osmosis is? Water is moving towards particles. And I'll teach you more about this in class, but it literally, particles equals party. So where does water want to be? Where the party's at. So wherever the party is at, wherever there's more particles, water wants to go there. And albumins have a very large draw for water. So since albumins are in your blood, water wants to be in your blood, and that's why so much water is in your blood. So albumins play a very large role in osmosis. You must know that, you must know that, you must know that. There'll be some other proteins we learn about through the semester, like fibrinogen we'll be learning later in today's lecture. Another one called plasminogen we'll also be looking at in today's lecture. Other solutes, electrolytes, nutrients, respiratory gases, waste products, these are a very small amount of your total plasma concentration. Are they important? Hell yeah, right? Because this is glucose and fatty acids. This is sodium and potassium and chloride and calcium, these electrolytes. So, oxygen, carbon dioxide, and then waste products like urea and many others, ketonic acids and stuff. So there are definitely other things, but they don't account for much of the total volume of your blood. I just spent a bit of time on this slide. Therefore, make sure you look at this slide. Make sure you understand this slide. Next, all of these formed elements, these blood cells, so to speak, they go through homeostasis. That's not the word you guys see here. Homeostasis, which means that if the body needs more of a particular type of blood cell, it will make it. Does that make sense? For instance, the function of your erythrocytes is to transport gases and a few other things. But what if we need to transport more gases? What are we going to do to the number of erythrocytes that our body produces? Increase, decrease, no change? Well, the answer would be increase, right? We need to transport more gases, therefore we need more erythrocytes. So the body is going to go through a process called hemopoiesis. Hemopoiesis, hemo meaning blood, poiesis meaning to grow or generate, kind of like a genesis kind of word. Hemopoiesis. Well, hemopoiesis all starts with a single stem cell line called a hemocytoblast, otherwise known as a pluripotent hematopoietic stem cell. You guys don't need to know that word, pluripotent hematopoietic stem cell. You guys can just learn it as hemo, blood, cyto, cell, blast, meaning to grow. Hemocytoblast is a blood stem cell that can grow into any formed element. So this is a really important stem cell line. And guess where it's found? Only location in the body where we find hemocytoblasts is in the red bone marrow. So wherever you have red bone marrow, that's where we find this hemocytoblast, which is really important because wherever we find hemocytoblasts is where we produce new blood cell lines, whether they're white blood cells, whether they're thrombocytes or erythrocytes, it doesn't matter. They're all formed in the red bone marrow. So make sure you ask yourself, where are all blood cells produced? Red bone marrow. So let's start walking through some of these lines. Let's learn more about erythrocytes, and let's learn more about leukocytes, and let's learn more about thrombocytes and what sort of things would cause them to be increased or decreased. Before we do that, I said it in the last slide, but where are all blood cells produced? In the red bone marrow. Okay, in kids, almost all of their bones have red bone marrow, and that's cool because kids are growing. Adolescents, children, so forth, so on, right? They have red bone marrow through all their spongy bone, pretty much all, and the medullary cavity of all their long bones. Cool. That's a lot of red bone marrow, and that's a lot of production of new blood cells, important for growth. Adults, however, once we go through puberty, a lot of our red bone marrow goes away. It turns into yellow bone marrow at that point. So most of those medullary cavities of the long bones that had red bone marrow when we were a kid, now as an adult, bummer. They're yellow bone marrow. We still have some locations for red bone marrow. This is important because if you're ever going to try to collect a patient's marrow, these are the sorts of locations that you're going to be collecting their red bone marrow from. So this is also if they have damage to any of these portions of bones, then you can actually have, think about it, right? If you don't have appropriately functional red bone marrow, then you're not going to be producing the proper amount of blood cells. So pretty cool to think about as a potential disease. All right. The first formed element that we're going to end up talking about is the most abundant of the formed elements. So why do we call these formed elements and not cells? You know, I've kind of been switching between blood cells and formed elements. They are technically called formed elements and not cells. And the reason why is because these blood cells don't act like typical blood cells. For instance, red blood cells, when you look at this slide here, they're the ones that look like little inner tubes, right? They kind of look like they're inner tubes floating down a river. It's the way I think about them, with the solid outside and the you know, almost clear inside, or you know, little donuts floating down a river. But inner tubes is cool. They don't have a nucleus, and so they're not your typical cell. They don't have a nucleus. They don't have mitochondria. They don't have a lot of the organelles that make a cell a cell. These little purple dots that just look like, I don't know, garbage. Well, those are thrombocytes. Okay, they don't have a nucleus. They're actually pieces of a bigger cell, and that cell actually cleaves pieces of itself off in the bloodstream and sends out these little thrombocytes throughout your blood. Crazy. And then the white blood cells, they do have a nucleus, but we want to combine all these things as a term. We end up just using formed elements. So the first formed element I want to talk about are erythrocytes. You may or may not know this, but erythro means red in Latin. Cytes is cells, so red cells. That's literally what the name means. And for the lab practical, you must write erythrocytes, and there are a lot of them. There are a lot of them. 5.1 to 5.8 million red blood cells per millimeter cubed. Per millimeter cubed. That might not mean anything to you, but basically a millimeter cubed is 1 ml of solution. 1 milliliter of solution. 1 milliliter, that's 1/1000th of a liter, and you have 5 to 6 liters of blood in your body. There are 5 billion cells per mil. Multiply that by 1,000, and that's how many cells per liter you have, and then multiply that by an additional 5. And you know, I haven't done that math in a long time, but it's a big number, right? 5 billion multiplied by about 5,000. Cool. That's how many cells, erythrocytes you have in your body. Okay, number one cell in your body. Most abundant, I guess I should say. And they're constantly going through a cycle of about 3 to 4 months. You're making new ones, old ones are dying. Your spleen and your liver are picking the old ones out, getting rid of them, and telling your body to make more. So what's so cool about erythrocytes? One, they're biconcave. This is important because the more surface area, this isn't flat, it's curved, right, on both sides. The more surface area that a piece of material has, then the more area something has to bind to it. So as we make a red blood cell biconcave, looking a little bit like an inner tube, right, or a donut, we increase the area that oxygen and CO2 and hydrogens and other things can bind to this red blood cell. Really cool aspect of the red blood cell. It's packed full of a molecule called hemoglobin. There's about 280 million hemoglobin molecules in one red blood cell. That's a lot of oxygen that it can transport. Crazy, crazy. Some other unique properties of these erythrocytes. Anucleated means that it has no nucleus, it has no mitochondria, because we don't want a cell whose job that is to carry oxygen. We don't want that cell to use it. So we don't give that cell the capability of using that oxygen. Kind of mean, but that's what it do. Some other properties of erythrocytes that are cool is it's very, very elastic. All of the cytoskeleton that holds this red blood cell together is basically just big old elastic rubber bands, which is important because when you get down into the capillary beds, which is the tissues of the body, this is where gas exchange occurs. These capillary beds are so small that only a single red blood cell can go through it at a time. And if they're about 5 microns wide, the capillaries, these blood vessels, and the red blood cells are approximately 7.5 microns, well, the blood vessel's smaller than the red blood cell. So these red blood cells need to squeeze down to get through that space. So they need to be elastic to do that job. If they lose that elasticity, what's going to happen to your body's ability to transport oxygen and CO2? Well, crap, now we're not being able to squeeze down through our capillary beds, no gas exchange or less gas exchange equals disease. What's going on with these hemoglobin? Hemoglobin are, again, I can't remember the number, 280 million hemoglobin per red blood cell. 280 million hemoglobin per red blood cell, multiplied by the 5 billion red blood cells per mil, multiplied by 5,000. You're starting to get the point. There are a lot of hemoglobin in your body. Hemoglobin are made up of alpha chains, 2 alpha chains, and 2 beta chains. And in the center of each one of these chains, these globin groups here, in the center of each one of these is this little disc shape called a heme group. So there are 1, 2, 3, 4 heme per hemoglobin. How many heme per hemoglobin? 1, 2, 3, 4. And in the center of each one of these heme groups, in the very center of each one of these heme groups, is an iron. And when oxygen binds, we create a specific conformation of the hemoglobin, and when oxygen is released, the conformation changes ever so slightly. When oxygen is bound, red. When oxygen leaves, a bluish color. Oxy versus deoxy, oxy versus deoxy, oxyhemoglobin, deoxyhemoglobin. What is this hemoglobin good for? It's good for transport. It's a transport protein. It's a carrier protein. It helps carry things through the bloodstream. So what is it helping to carry? Well, it depends on what part of the hemoglobin we're talking about. The heme portion or the alpha beta chains, which are called globins or globins, but globin is how I was learned, how I was taught. All right. Oxygen, carbon dioxide, glucose, carbon monoxide, EGADs, very poisonous, hydrogens, nitric oxide, all of these can be carried by the hemoglobin, but which portion is important? Oxygen and carbon monoxide are both carried by the iron. They're carried by the heme. The center portion of each one of these groups carries oxygen and carbon monoxide. The globin itself, this big raveled protein, this binds carbon dioxide, glucose, hydrogens, and nitric oxide. So everything else binds to these globin chains, which is important because if carbon dioxide binds over the heme group, slightly bigger, so it will block oxygen from being able to bind to that heme group. We'll talk more about carbon monoxide when we get to the respiratory system, but for now, let's skip by it. All right, what if we want to produce new red blood cells? We need a process for this. And the body has this beautiful homeostatic regulatory function for producing red blood cells. Erythrocytes. The formation of new erythrocytes is called erythropoiesis. Remember hemopoiesis a little bit earlier? New blood cells. Well, this is specifically new red blood cells called erythropoiesis. And the hormone that's involved in this function is called erythropoietin. Proteins that end in IN are functional hormones, functional protein hormones. So if they end in IN, they're actually not even a hormone, they're just functional proteins. So you guys learned about a lot of proteins that end in IN and AMP1 with, you know, actin, myosin, tropomyosin, troponin, titin, nebulin. So just in skeletal muscle alone, there was a whole bunch of proteins that end in IN. You'll be learning more this semester. So the way this all works is relatively simple. You know, let's see if I can get this thing up and running without. Oh, it shows me. Why not have this on a recording for the rest of my life? Super scrubby day. And now it's recorded. All right. So you guys know I have to turn on and off my machine to get this thing to read sometimes, and I did not do that beforehand. But so the way this all works is relatively simple. Okay. And we know that the body is all about stimuli. We know this, that a stimulus, it can talk to a receptor in the body, wherever that receptor may be. And then that receptor has a function. It's going to have a response. True story? I mean, we're kind of, this is physiology in general, and the response is always trying to help fix the problem created by the stimulus. We often just call this a negative feedback loop, right? So this is the simplest idea of these homeostatic loops. The stimulus in this case that causes the production of new erythrocytes, well, I think the easiest way to do this is to try to remind ourselves what is the function of erythrocytes. What is one of the primary functions of erythrocytes? It's to carry oxygen. So what better stimulus for the production of new red blood cells than a change in oxygen levels? So if a patient, human, starts to have an increase in oxygen demand for whatever reason, or if there's a decrease in available oxygen, I hope it stands to reason that in either one of these cases, the body is going to want more oxygen. It's going to be like, I have an increase in metabolism. I need more oxygen. Or, man, there's not enough oxygen available for my body to do what it needs to do. I need more oxygen. Well, the body, sure, is going to increase its respiratory rate. Awesome, great. However, that doesn't mean oxygen is getting out to the tissues, because oxygen is extremely nonpolar. Oxygen needs erythrocytes and the hemoglobin and the heme group to get to where it needs to go. So when I have an increase in oxygen demand and a decrease in oxygen availability, either one of these, we need more erythrocytes to help with the transport of oxygen in either one of these cases. We can't just breathe faster. That doesn't do crap if we don't have transporters available to move that oxygen around. So what we have is an organ with receptors in it for oxygen. This organ is your kidneys. So as oxygen demand goes up or oxygen availability drops, the kidneys can recognize this. The kidneys are great for this role because the function of the kidneys is to filter your blood. In fact, there are 180 liters of blood being filtered by your kidneys every day. Approximately every minute, 20% of your blood is filtered. Well, that's a lie. Approximately every minute, 20% of your blood goes through your kidneys. How about that? So every 60 seconds, approximately 20% of your 5 liters has passed through your kidneys. So what better organ for trying to monitor blood than those kidneys? So kidneys detect this change, and the kidneys say, let's fix this problem. How do the kidneys do that? The kidneys synthesize, they produce a hormone called erythropoietin. And we abbreviate erythropoietin as EPO. So the kidneys have already synthesized EPO. Capital E, capital P, capital O stands for erythropoietin. So this is a protein. It's already been synthesized by the kidneys, and it's sitting there waiting in vesicles. And so as the oxygen demand changes or oxygen availability changes, the kidneys can either release more erythropoietin or less erythropoietin, depending on how much oxygen is there. But if we have an increase in oxygen demand, we need more erythrocytes. So what this is going to do is it's going to cause the kidneys to secrete EPO into the blood. Does that make sense? I hope so. This erythropoietin is a hormone, and it is now going to go to another target organ. What makes a target organ a target organ? Receptors. So where are the receptors for EPO? All right, logically, let's think about this. What are we trying to make more of? We're trying to make more blood cells. We're trying to make more erythrocytes, red blood cells to be more specific. And where are all red blood cells produced? Red bone marrow. So where are the receptors for erythropoietin? In the red bone marrow. So what are we going to do? We're going to draw arrows out. We're going to say this is going out to the red bone marrow. And that erythropoietin acts on the red bone marrow, starts to stimulate those hemocytoblasts, pluripotent hematopoietic stem cells. Then that's going to increase the synthesis or production, increase production of new erythrocytes. Again, this is called erythropoiesis, erythropoiesis. This is the whole category here. EPO stands for erythropoietin. And they're going to help make new erythrocytes. And if there's more erythrocytes, now we can carry more oxygen. Just as a side note, EPO is one of the regulated drugs from the Olympic Committee and from lots of other committees as well for the NFL and whatnot. Athletes are not allowed to take EPO. That's considered doping. They can literally just take EPO. In fact, patients that have undergone chemotherapy, patients typically with cancer that have undergone chemotherapy, what's happened to their number of total cells in their body? Well, chemotherapy goes through and kills cells, including a lot of your blood cells. And so they will give those patients EPO after treatment to try to bring their blood cell production back up again, red blood cells to be specific. So yeah, there's a whole bunch of real life scenarios for us to understand erythropoietin a little bit better. So let's do one more quick case study. Everybody just stop and think. You go to climb a mountain. In case you don't know, as you increase in elevation in the world around you, there's less oxygen available for you to breathe. The air is quote unquote thin. And if there's less oxygen in the air around you, you have a decrease in oxygen availability. And therefore, you get off the plane in Salt Lake City or whatever, go to climb a mountain, and you're panting and breathing super hard, and you're lethargic and tired because there's just not enough oxygen available. So what's your body going to do? Well, crap. You get off that plane in Salt Lake City, you're breathing hard, your kidneys detect that drop in oxygen. It starts to secrete erythropoietin. That erythropoietin gets secreted into the blood. The red bone marrow starts to produce new erythrocytes. Now, is this immediate? No. Production of new erythrocytes takes days, typically at least 3 days. But if you're in Salt Lake City for longer than 3 days, all of a sudden you don't feel so tired. You're not panting as you're moving around, hiking, skiing, whatever you may be doing up there. Pretty cool. When you climb a mountain, base camps like for Mount Everest, you got to be at those base camps for 3 days. Why? So that your body produces enough new erythrocytes to be able to transport the oxygen that you're going to be given. So lots of real life examples with EPO. Pilots have high EPO all the time. Why? Because pilots are always dealing with these pressure changes in the air around them. So pretty cool. All right. Recycling and elimination of RBCs is a pretty big deal. And this flow chart's going to be really tough for you guys to read from here. So here's where I'm going to ask you guys to really sit down and walk your way through at least the gist of recycling and eliminating RBCs. Remember, this happens every 3 to 4 months. These erythrocytes, if they get old, they cannot go through your capillary beds properly. So these aged erythrocytes, they've lost their elasticity, they're dead, they're just starting to not be able to transport oxygen very well. Well, they're going to be detected by either the liver or the spleen, and they're going to begin to be broken down. And as they get broken down, we got to think about the different parts of the erythrocyte. Okay, the majority of the erythrocyte just gets eaten up, but what's left over is the hemoglobin. And it's the hemoglobin that your body kind of has to figure out what it needs to do with. What is that hemoglobin made up of? 4 globin groups, 2 alphas, 2 betas, a heme, and in the center of the heme there was an iron. So as that erythrocyte is degraded in the liver/spleen, we end up with globins, irons, and a heme that does not have the iron anymore. Globins, recycled, awesome. We take these, pretty easy to break down proteins in our body. We've got all sorts of enzymes that help us do that. They get broken down in individual amino acids, and then those amino acids are reused by the body. Sweet. Gluten's pretty easy. Iron. Iron's a lot more challenging because iron's a metal, and it's kind of tough to move around. We have some hormones to help move it around, but iron can be lost from the body in many different ways. Most of our iron is stored in the liver. Some is actually going to get lost, though. Iron loves to go out in your sweat, pee it out, and then, of course, if you're bleeding, menstrual cycles, that sort of stuff, there's a lot of different ways that iron can be lost. Bad news bears. Bad news bears. Therefore, we always have to be consuming some level of iron in our diet. Don't forget that. You got to have iron in your diet, otherwise you become anemic. You could produce as many erythrocytes in the world as you want, but if there's not enough iron to build your heme groups, then those erythrocytes tend not to be able to carry as much oxygen. All right, the most challenging part of all of the breakdown of erythrocytes is the breakdown of heme. Heme is broken down via a very complex process. It's as simplified as possible in this particular diagram here. But the heme group has to get broken down into a number of chemicals that are important. Many of you have probably heard of before. Biliverdin, you probably haven't heard of that one before, but biliverdin has to be converted from heme in order for bilirubin to be produced. And bilirubin is a metabolic waste. Bilirubin is a product of heme breakdown. And bilirubin is this yellowish, greenish, more yellow-orange kind of color, bilirubin. And it's excess bilirubin that ends up causing jaundice. Kind of a cool idea, right? Bilirubin, if it builds up in the body, causes that disease called jaundice. And almost every baby that's born has jaundice. Why? Because that baby is right in the process of converting mom's red blood cells and producing their own red blood cells. So that those babies, those newly born babies, are breaking mom's red blood cells down and producing their own red blood cells, but they're doing it so fast that they can't eliminate all the bilirubin fast enough. They're breaking down the erythrocytes in the heme group so fast that they can't eliminate it. And so it ends up building up in their bodies. It builds up in their livers, in their digestive system, in their kidneys. And if it starts to build up, then it starts to push back. It ends up in the skin and in the eyes. And that's what makes those babies' skin so yellow when they're born in. Pretty cool. Bilirubin, just kind of, when you guys go to study this, make sure you follow this flow chart where it's transported to the liver. What's going to happen to it in the liver? How does it end up going through the liver? By albumin, you know, that transport protein that actually helps with osmotic potential. Well, it's moving that bilirubin around. Now, what's going to happen to it is it's going to end up getting transported from there into the intestines, and it does that through bile. And then it gets converted to urobilinogen, and then you end up excreting some urobilinogen and stercobilin in your feces, and then some of the urobilinogen ends up in your urine, and it's a big crazy mess. Now, I won't test you on the entire flowchart, I promise, but I will potentially test you on bilirubin buildup, and make sure you understand that if more red blood cells are getting broken down, that's going to result in more bilirubin. That's the end of the erythrocyte abbreviation. Whew. White blood cells. There are a lot less white blood cells in the body. 4,500 to 11,000 per millimeter cube per mil, in essence, is what you're looking at. These guys right here are immune cells. They're not just any immune cells, they are the immune cells of the body that are motile, which means they move. They can move through the bloodstream and they can move through the tissues. They have this really cool process called diapedesis that allows them to move through the tissues into damaged tissues. Cool. That's about all I want you guys to know about the general function of leukocytes. Now, there are 5 major types of leukocytes. You've learned these for lab, or will learn these for lab. There are neutrophils, eosinophils, and basophils, and they're all called granulocytes because when you look at them under a microscope, they have heavy granulations. They look like pieces of sand. And there are lymphocytes and monocytes, and these are called agranulocytes, and A in Latin means without or none. So agranulocytes tells us there are no granules in lymphocytes and monocytes. And if you look at the cytosol, you won't see granules. Cool. Make sure you guys know these range values for the number of white blood cells. Please, please, please. Of course, my wife is calling me right now. 1 second. Okay. I always love distractions while I'm trying to do a recorded video, but they happen. So you need to know these range values. You got to. Now, there is a way to be able to memorize most abundant to least abundant, and it's with a mnemonic that goes, never let monkeys eat bananas. Never let monkeys eat bananas. And that's going to let you know the order of most abundant to least abundant. This will be important because if it's the most abundant, probably means, remember this is immune system, it probably means that it's fighting something that's pretty common. Whereas if it's the least abundant, it probably means that you don't have that much need for it most of the time. Does that make sense? I hope so. Remember, the formation of new red blood cells was called erythropoiesis because it was making new erythrocytes. So now the formation of new white blood cells is called leukopoiesis because it's making more leukocytes. And you don't need to know most of this crazy mess that's here. I won't test you on it. However, you do need to know that in order to get new white blood cells, these hemocytoblasts are going through these little differentiation points here. But you'll notice that the thing that they have in common is this funny word called CSF. CSF. So in order for a white blood cell to become a white blood cell, it needs this, it's called a GM-CSF, but it's a colony stimulating factor. Colony stimulating factor. And that's one of the things I might ask a homework or test question on. Is if I have colony stimulating factor, will I be producing erythrocytes? No. Colony stimulating factor, especially the GM colony stimulating factor, is related to leukocytes. And you can have all of these different byproducts, whether it's the granulocytes, the monocyte, or from the lymphocytes. There are multiple types of lymphocytes, B lymphocyte, T lymphocyte, and natural killer cells. All of these can be produced through leukopoiesis. In this summary here, I have a very detailed description of this already on Blackboard for you guys, so please listen and watch and take notes on it from there. This table is a fantastic one, so pause it now if you'd like, and make sure you understand the characteristics that would allow you to identify these via histology. But more importantly, make sure you understand their function and their approximate percent. Remember how I kept on saying neutrophils are the most abundant. Neutrophils are the most abundant. They make up 50 to 70% of the total number of leukocytes. Why is that? Because they are fighting the most abundant thing in the world around you. Take a breath in. touch your skin, touch the desk in front of you, wherever you may be, there's bacteria everywhere. And bacteria are a patho. They are something foreign to your body. So if it gets into your body wants to fight it. What do you guys have in place to fight bacteria? Neutrophils. So if my range is different than normal, think about it. If you got a patient that comes in, you take their blood, and their neutrophil count is 80%, what do you know they're fighting? A bacterial infection. Eosinophils are all about parasites, parasitic worms and stuff, parasites, and then allergies. So what if somebody comes in and their eosinophil count is 6%, 7%? You know they're either having an allergic reaction or they're fighting a parasitic infection, a parasitic infestation. Basophils, their function has to do with inflammation. And they release histamine. You guys have ever taken an antihistamine, you know that it fights inflammation. Well, perfect. Basophils are all about inflammatory reactions. And so if somebody comes in and their basophil count is like 2%, that's a big number, you know they're having some sort of inflammatory reaction. Lymphocytes. These are the rest of the pathogens, really, that the body's trying to fight. Viruses, infections, COVID. Okay, any pathogens that really come into the body that are abnormal. And they could even help fight bacteria, by the way. But really anything foreign to the body, the lymphocytes produce the antibodies. Remember your blood typing information? Okay, the lymphocytes are what produce the antibodies, and they tag the pathogens for removal. So again, if somebody comes in and their lymphocyte count is 60%, 50%, then you know that they're having some sort of viral infection, most likely a viral infection or some other sort of pathogen infection, maybe even like a sliver. Maybe they got a porcupine quill inside of them, you know, they're attacking these pathogens. Once they're tagged with an antibody for destruction, that's what the monocyte's job is. So the monocyte goes around and it phagocytizes all of those pathogens that have been tagged for removal. So these monocytes are going to be able to, not in and of themselves, detect pathogens, but with the help of lymphocytes, they can detect pathogens and eat them. Phagio means to eat. Cytize means to internally eat inside the cell. So there you go. Now, if somebody's monocyte count is high, that means that they are eating lots of stuff. Cool, I hope that all helps. The last formed element is the thrombocyte. We're going to spend the rest of this summary talking about thrombocytes. Thrombocytes are all about clotting. Okay, there's your normal values, 150 to 400,000 per millimeter cubed. If this value is too high, that means they have